Hi, Miss Cecilia here, and in today's episode of Bio Mini, we're going to talk about one of the central topics in understanding biology. With that, let's start. Francis Crick, who was the one who coined the term central dogma, said, "I call this idea the central dogma for two reasons, I suspect. I had already used the obvious word hypothesis in the sequence hypothesis, and in addition, I wanted to suggest that this new assumption was more central and more powerful." So with that, what is the central dogma of molecular biology, and why is it such a central concept in understanding life? A gene that encodes a polypeptide is expressed in two steps. In this process, information flows from DNA to RNA to protein, a directional relationship known as the central dogma of molecular biology. Your DNA, or deoxyribonucleic acid, contains the genes that determine who you are. But how can this organic molecule control your characteristics? Well, DNA contains instructions for all the proteins your body makes. Proteins, in turn, determine the structure and function of all your cells. However, what determines a protein structure? It begins with the sequence of amino acids that makes up a protein. Instructions for making proteins with the correct sequence of amino acids are encoded in your DNA. DNA is found in chromosomes. In eukaryotic cells, chromosomes always remain in the nucleus, but proteins are made at ribosomes in the cytoplasm. So how do these instructions in DNA get to the site of protein synthesis outside the nucleus? Another type of nucleic acid is responsible. This nucleic acid is the RNA, or ribonucleic acid. RNA is a small molecule that can squeeze through pores in the nuclear membrane, and it carries the information from DNA in the nucleus to a ribosome in the cytoplasm and then helps assemble the protein. So in short, we have the flow DNA to RNA to protein. Earlier, we mentioned that this process takes place in two steps, and those two steps are transcription and translation. Transcription is the process by which DNA is copied to mRNA, which carries the information needed for protein synthesis. Transcription takes place in two broad steps. First, pre-messenger RNA is formed with the involvement of RNA polymerase enzymes. The process relies on Watson-Crick base pairing, and the resultant single strand of RNA is the reverse complement of the original DNA sequence. We will learn more about this in our classroom discussion. Now, the pre-messenger RNA is then edited to produce the desired mRNA molecule in a process called RNA splicing. Translation, on the other hand, is the process wherein the mRNA is formed in transcription, is transported out of the nucleus into the cytoplasm to the ribosome, and here it directs protein synthesis. Messenger RNA is not directly involved in protein synthesis, but rather transfer RNA is required for this. The process by which mRNA directs protein synthesis with the assistance of tRNA is called translation. And with that, we have learned the basics of the central dogma of molecular biology. This has been Miss Cecilia, your host for this Bio Minis lecture. Till next time!